Leading a startup team, whether you're delivering a sugar rush, stocking coffee, or getting a regular delivery of snacks, Office Depot has solutions that fit every startup culture, from getting those first business cards and stationery to ordering fleece pullovers with your new logo. To learn how Office Depot and the California Technology Council have partnered to bring you savings on all of these startup essentials and more, go to californiatechnology.org/member benefits. I'm Daniel Levine, and this is the Bio Report. Acepodia has developed platform technology that allows it to chemically modify or conjugate living cell surfaces. The technology can be applied to any immune cell and any antibody or binding protein without the use of genetic engineering. The company is working to apply the technology to create a family of cost-effective, off-the-shelf immunotherapies. We spoke to Sonny Hsiao, CEO of Acepodia, about the company's platform technology, how it works, and why it has the potential to change the cost and efficacy of immunotherapies. Sonny, thanks for joining us.
Thank you.
We're going to talk about Acepodia, its efforts to develop affordable immunotherapies, and your ACC technology that allows you to modify or conjugate living cells. Let's start with the state of immunotherapy and cell therapy in cancer today, though. What do you see as the biggest challenges with existing approaches today?
There are two challenges for the cell therapies in immuno-oncology today. One is the ability and potency to fight solid tumor, or we say to overcome the solid tumor microenvironment. And second is to provide an affordable treatment so that the cost of good and then also the affordability can be better so that everybody can use.
What's the potential to deliver off-the-shelf therapies, and what impact might that have on the cost and expanding the use of these therapies?
So the off-the-shelf therapy that Acepodia is developing, it will be much simple and straightforward in comparison to the current autologous therapy. Autologous therapy meaning that it will need to get the blood from the patient from the hospital and then ship that blood to another facility. Take 2 to 3 weeks to make the product at the facility. One patient, one product, and then ship it back to the hospital. So you can see it's a very lengthy process and a complicated process. So based on the estimation from the academia and the industry, the off-the-shelf therapy will be about 100 times more cheaper than the autologous therapy.
What exactly is your ACC technology, and how does it work?
Our ACC technology was the work I developed in UC Berkeley. ACC stands for antibody cell conjugation. So it's using a bioconjugation technology to link the antibody and cell together. So it can unite antibody therapy and also cell therapy to provide a very potent treatment. And then another strength of what we do, the ACC, is a bioconjugation process. So it doesn't require any genetic manipulation of the cells. It will just be a simple conjugation step bioconjugation meaning that you link one biomolecule to the other using the chemistry or biochemistry, so that the process is easy, and then also it provides a different and more potent mechanism for the cell to fight cancer. Because you can imagine conjugating an antibody to the cell, we call ACC, is kind of a missile guidance system. We put a missile guidance device, the antibody, which can target and then guide the cells towards the tumor, so that these missile guidance cells can bring our powerful immune cells to target tumor and then kill them.
And is the expectation that you would only use the antibody for targeting purposes, or could it be used for an additional therapeutic benefit as well?
So it can do both. On the top level, it is a guiding system, so that it provides the targeting of the NK cells, for example, natural killer cells, which is one of the powerful first-line defender of our immune system to target and then kill tumor cells. And then also the second level is that this antibody conjugating to the NK cells, when the NK cell engage tumor cell, antibody also help activate the NK cells and stimulate the NK cell to kill the tumor cells. So antibody in our ACC system does have two-pronged properties. One is the targeting. The other one is activating NK cells to kill the tumor cells.
I know one of the challenges with antibody drug conjugates has really been in the linker itself. How much of a challenge is getting the chemistry right to link an antibody to a cell?
So it is a very good question. So this is what we spent many years to develop this technology, because it is actually even more challenging than the ADC. As you can imagine, we are conjugating antibody to a living cell. So it is a living cell. So our conjugation per se cannot interfere with cell's own biochemistry. In a simple term, our chemistry cannot kill the cell. Okay, so actually 99.9% of the chemistry bioconjugation, the conjugation technology will kill the cell. So we're working on this 0.1% chemical conjugation we can use to conjugate antibody to the cell. So that's the technology. We have the patent from UC Berkeley, and then it is also take many years to develop this chemistry, because unlike ADC, ADC is an antibody drug conjugate. You're conjugating a chemo drug, a toxic chemo drug to the antibody, but that drug is not alive, right? The drug is dead. So to the ACC, we're actually conjugating antibody to a living cell, living organism, and then that chemistry will be even more challenging, because that chemistry cannot kill the cells.
You mentioned that you're using chemistry as opposed to engineering, cell engineering, to attach the antibody to the cell. What's the implication of that from a manufacturing point of view?
Yeah, so we are using the chemistry instead of genetic engineering. So the chemistry have two advantages for the manufacturer. One is that it's much simple and straightforward. So this bioconjugation we do, we have only two major steps. One is conjugating a linker on the antibody. The second part is conjugating another linker on the cells. So it's take a metaphor, it's like a Velcro system. Have a linker on the cells, have a linker on the antibody, and then we mix them, they will conjugate it to each other. So it's only 2 steps versus the genetic engineering, which has many, many steps, including the bead selection, transfection, transduction, virus clearance, because you need to use virus. So there are a lot of steps involved in the genetic engineering, and then also a lot of the assays and then the steps you need to do to make sure the virus got cleared. However, for us, there's no virus touching the cells. There's no virus in the manufacture, so that the process is very simple, very safe, and then also can be scaled up easily.
I suspect one of the other advantages, you could move fairly fast preclinically in going from a desired target to an end product.
Yes. So so that advantage of the manufacturer, we're talking about simple and quick, that also applies to the R&D, because intrinsically making this product is much simple and quicker, so that we were able to shorten the development time of the cell therapy, and then if we have a desire and validated antibody, we can make the product in a relatively short time to expand our pipeline.
And what kind of cells are you using? Is one type of immune cell preferable to another?
So currently the ACC technology itself, it can be applied to a variety of the cells. For example, the cells we tried, including NK cells, natural killer cells, T cells, and also cytokine-induced killer cells, which we had just our collaboration with Stanford University just published, and then also can even apply to dendritic cell or stem cells. So the application is really broad. However, currently, for the company perspective, we are focusing using the NK cells right now, and then the reason we're picking the NK cells as the first candidate is because NK cells can be used as an off-the-shelf treatment, and then also NK cells have very good synergy with antibody, meaning that antibody can activate NK cells relatively easy, and then give them power to fight cancer.
Let's talk about your pipeline. ACE-1702 is your lead therapeutic candidate. This is in development for HER2-positive cancers. What exactly is it?
So 1702 is our currently the first product in our pipeline. And then as you can imagine, the concept of ACC, we conjugating an anti-HER2 antibody to the NK cells, natural killer cells, to fight solid tumor. So anti-HER2 is the biomarker expressing variety of solid tumors, including breast cancer, ovarian cancer, endometrial, gastric, and lung, etc. So it's a tumor biomarker, and then so what we do back is this missile guidance system to get the anti-HER2 antibody, which can target the tumor expressing this biomarker HER2, and then get the NK cells equipped with this missile guidance system so that they can go and then kill those tumors. And then this 1702, we just recently in January got the FDA clearance, and then now we're starting our trials in 2 or 3 medical centers in U.S.
I imagine that there was no shortage of indications you could have started with. What was the thinking in choosing to start with HER2-positive cancers?
So correct. So on our pipeline, and then as we discussed just shortly before, we actually can expand our pipeline relatively easy and quicker. And then we do have our pipeline developed and also listed on our website. It will be HER2 followed by PD-L1 followed by CD20, CD19. So it is not limited to HER2. However, HER2, what we're picking HER2 is that HER2 is a well-established tumor marker. And then although there are current drugs, however, there are still more than 50% patient relapse. So and then some of the indications, for example, ovarian cancer, endometrial, those women cancers, the tumor cells express HER2. However, current HER2 drug, the existing drug, haven't got approved yet. So we hoped our supercharged NK cells 1702 can provide another and then hopefully better treatment for these patients.
What's known about 1702 from the preclinical work that was done?
So 1702, we have done the preclinical work both in vitro and in vivo. That meaning we have done the experiment studying 1702 at the cell level, that's in vitro, to see if they can kill the tumor cells when we mix them directly with the tumor cells. That's the in vitro study, and it worked. And then also, second, we also did the in vivo study, which meaning that we applied the 1702 to the tumor-bearing mouse. So those tumor mouse, they have tumors, they develop tumor, and then we apply 1702 to those mouse. They worked as well. They show good efficacy. And then the third, which is even more deeper in science, is that we also did we call tumor microenvironment study. So the current challenge, as I said in the beginning, is that for the solid tumor, when the tumor develop, they also develop a suppressive microenvironment, including low oxygen environment, high acidic environment. So those environments are adverse to the immune cells. So that's why a lot of time, currently, for many indications of solid tumor, there's still no cure. And then we also go ahead did a preclinical study, studying what happened to our cells under those suppressive microenvironment. For example, low oxygen, and then also, for example, more acidic, which is low pH. And then under those conditions, in the preclinical study, we also see our 1702 work quite well, even under those suppressive tumor microenvironment.
Well, given the ability of the microenvironment to suppress the activity of an immunotherapy, we've seen a lot of people moving towards combination therapies. Is the expectation that your cells would be used as monotherapies, or would they be used in combination with other agents?
So our cells will be first tested as a monotherapy, and then in animals we see that as a monotherapy, it does have good efficacy. So that's we will pushing it forward. However, this is based on the current study from our lab, and then also from the academia and some industry, is that if the cell therapy can also be a good combination with other drugs as well. For example, chemotherapy, because the chemotherapy can first debulk the tumor. However, chemotherapy usually cannot wipe out all the tumor cells. On this part, cell therapy coming in will bring the advantage to kill and then trace down the metastatic tumor cells. So again, for us, we go step by step. We do the monotherapy as we see good efficacy in mouse. However, once we establish that cell therapy does have the potential to combine with other treatment or standard of care as well.
I also wanted to ask you about your other experimental therapy, ACE-1708, which is actually a combination of NK and T cells. What's the thinking in combining the different cell types?
So this I want to clarify a bit. So 1708 is not it's not combining us, meaning that we put both NK cell and then T cell in, is that our 1708 is conjugating an anti-PD-L1 antibody to the NK cells. However, this anti-PD-L1 antibody is to some extent even more powerful because it serves two purposes. So anti-PD-L1 antibody, first, it can direct NK cell, guided NK cell towards the PD-L1 expressing solid tumors. And then second is that upon binding the PD-L1 will also trigger the surrounding T cell to be activated to attack tumor as well. So that the combination effect as seen here is that although we get the anti-PD-L1 conjugated NK cells, which is our 1708, it can do two things. One is that it can target tumor cell and kill the tumor cell. Second, it can activate the endogenous T cells, meaning that the T cells already in our body, and then to fight as well. So it is an exciting product as that it can bring in both the T cells and then NK cells ability to fight cancer.
You're starting the clinical program at a time where we're all wrestling with the COVID-19 pandemic. Has that impacted the ability to conduct the trial in any way?
So this is a very, very good question. So the COVID-19, the coronavirus, as we all see, does have the impact in our healthcare system. So that I think the first thing we do for the clinical trial is that we hope, and then also we send out a letter to our site that the patient who want to participate in our trial will have the test first, if possible, and then we think it is possible for our trial because now we have more and more testing kits that's available, and then especially when those patients participate in the trials, they're coming in the hospitals many times to receive the drug and then do the testing. So it would be better to have them tested first, so that they can know they are safe. The doctors will know they are safe. So I think that's the measure we'll take for the clinical trials. We're going to start, and then that's kind of from a sponsor perspective.
I know you're focused on cancer. I'm curious though, do you see this technology having implications outside of cancer?
Yes. So the cancer is our current focus right now, and then we believe with this missile guidance, the targeting system ACC, we do have the ability to provide a different and better mechanism to fight cancer. However, there are many other indications we can potentially target as well. For example, fibrosis. In this indication, for what we do, instead of killing the tumor cells, we're going to kill those bad fibroblast cells, which cause the fibrosis. So as you can imagine, it's still the same concept, right? So patients with NASH, with fibrosis, they develop these bad fibroblast cells in their body. So that if we have a good antibody can target those cells, we can use ACC and then conjugate those antibodies to the NK cells. And then now these antibodies can bring the NK cells to kill those bad fibroblast cells, and then hopefully can provide a treatment for the fibrosis patient.
Sonny Hsiao, President, CEO, and co-founder of Acepodia. Sonny, thanks so much for your time today.
Thank you very much, Daniel.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.